This video was made possible by Thunderbird Perch. For generations, the story of losing one's mind has been a story of bad luck. We watched loved ones fade, their memories and personalities slipping away. We chalked it up to the cruel lottery of genetics or the inevitable decay of old age. The official narrative offered little more than sympathy and a shrug. Neurodegeneration was seen as a tragic but random event, a lightning strike from a clear sky. But what if that story is incomplete? What if the sky is not so clear after all? Emerging science is asking a profoundly different and more unsettling question. What if our modern environment is methodically disabling our brain's most critical defense system, leaving it vulnerable to a slow internal corrosion? At the center is an enzyme known as GPX4, the crucial line of defense whose failure points to a measurable, potentially preventable process. To understand the danger our brains are in, we must first appreciate the guardian that stands in the way. GPX4, glutathione peroxidase 4, is a microscopic marvel of biological engineering, a specialized maintenance worker inside your brain cells. Its job: patrol delicate, fatty membranes, the walls, gates, and communication lines of the neuron, which are incredibly vulnerable. Packed with polyunsaturated fats, membranes are like a tinderbox waiting for a spark. That spark is reactive oxygen species, ROS, which can trigger a chain reaction of damage. GPX4 neutralizes lipid peroxides, oxidized fats that can propagate destruction across the membrane. Using glutathione as its fire extinguisher, it converts lipid peroxides into harmless lipid alcohols, stopping the cascade. Without functional GPX4, ferroptosis can be initiated. This isn't just any form of cell death. Unlike clean, contained programs, ferroptosis is messy. Membranes disintegrate, contents spill, and inflammation is triggered. In the tightly packed brain, one neuron's ferroptosis creates a toxic environment for its neighbors, a slow-burning fire through circuits. The result: large-scale tissue loss seen in neurodegenerative disease. The health of our brain is tied to this tiny enzyme, an active, vigilant protector, an anti-rust coating for brain machinery. Reduce the number of GPX4 enzymes, inhibit their activity, or prevent them from reaching membranes. Each is a direct threat to long-term cognitive health. How do we know for certain that the failure of GPX4 can cause neurodegeneration? In 2025, a groundbreaking study in Cell provided a smoking gun. Researchers identified a family with severe, early-onset dementia and brain atrophy. The cause: a single tiny mutation in the GPX4 gene. This wasn't environmental exposure. It was a blueprint flaw, an inborn vulnerability. The catalytic function remained intact, but a small fin-like loop, the membrane anchor, failed. The enzyme couldn't stay tethered where it was needed. The firefighters were ready and willing, but they couldn't reach the fire. Lipid peroxides built up on neuronal membranes, triggering rampant ferroptosis. The patient's brains began to shrink. Dying tissue showed protein aggregation and stress markers, matching what we see in Alzheimer's brains. This is proof of mechanism. A broken GPX4 shield leads to ferroptotic cell death, which in turn leads to the clinical picture of dementia. If one mutation can disable the shield, what happens when the environment delivers many smaller, cumulative attacks? The genetic failure of GPX4 is the exception. For most, the blueprint is fine. The emerging concern is that our modern world has created a new set of rules: the trifold attack. Chemicals, frequencies, and biological imbalances converging to weaken this defense in the general population. It's a theory of synergy. The whole is more dangerous than the sum of its parts. Each fold attacks from a different angle, but all target the delicate balance between oxidation and protection within brain cells. This model shifts focus from a single internal defect to multiple external pressures. It's the difference between a bridge collapsing from a sudden failure and a bridge weakening over decades from corrosive air, vibrations, and heavy load. Our brains are that bridge. Environment accelerates decay, with GPX4 failure and ferroptosis at the epicenter. The first fold is chemical, the second is energetic, the third is a biological breach, all pointing at GPX4. By categorizing threats, we can test pesticides on GPX4, frequencies on oxidative stress, and heavy metals on iron metabolism. The trifold model gives us a map. The first and most tangible front is chemical. Our biology never evolved for today's synthetic load. Two culprits stand out: agricultural pesticides and urban air pollution. We breathe them, absorb them, and ingest them. New research connects these exposures to the machinery that protects neurons from ferroptosis. Organophosphates are long-known neurotoxins. The EPA has moved to restrict several, citing serious health risks. Historically, we focused on neurotransmitters, but a new layer emerges. Some exposures may directly suppress GPX4 production. This is crucial. It means the pesticide isn't just interfering with signaling, it's dismantling the brain's defense against oxidative collapse. Even more pervasive is PM2.5, ultrafine particles that reach the lungs, bloodstream, and the brain. A landmark 2025 study showed neurons exposed to PM2.5 undergo a cascade aligned with ferroptosis. GPX4 plummeted. Iron and lipid peroxidation soared. Neurons began to die. Add ferrostatin 1 and neuronal death fell, confirming the ferroptosis pathway. Further research showed PM2.5 inhibits ERK-CREB, an on-switch for the GPX4 gene. In essence, pollution tells the cell to stop making its own shield, then floods it with oxidative stress. Multiple 2025 studies link environmental pollution to ferroptosis via iron transport, ROS, GPX4 dysregulation, and autophagy pathways. The second fold is more abstract: the saturation of man-made electromagnetic frequencies. Cell phones, Wi-Fi, towers, broadcasts, and countless wireless devices form a dense web of energy. Debate persists, but research is honing in on oxidative stress as a plausible mechanism. Low-frequency EMF can't break bonds like X-rays, but it can excite molecules and increase reactive oxygen species. These ROS are sparks GPX4 cleans up. In a healthy cell, manageable. In a compromised cell, dangerous. What if EMF raises ROS in a brain where GPX4 is lowered by pesticides or PM2.5? The defense is quickly overwhelmed. Lipid peroxidation accelerates. The cell crosses into ferroptosis. Large human studies are needed, but radiation research shows brain tissue can be driven into ferroptosis by high-energy waves creating massive ROS. Intensity differs from daily EMF, yet the pathway, energy, oxidation, ferroptosis, aligns, especially when defenses are down. The final fold comes from within: a biological breach. Accumulated heavy metals and chronic systemic stress can turn the internal environment hostile. They manipulate a key ingredient: iron. They load the gun, making membranes dangerously susceptible to oxidative damage, effectively mimicking conditions of a GPX4 genetic failure. Lead, mercury, manganese, and excess environmental iron can disrupt delicate brain chemistry. The brain locks iron in proteins like ferritin. Free iron is highly reactive and catalyzes ROS. When metals enter the brain, they can displace stored iron, increasing the free iron pool, priming ferroptosis. Chronic inflammation, lingering infections, poor diet, and autoimmune conditions flood tissue with inflammatory signals and ROS, adding to oxidative load. Even a healthy GPX4 system can be overwhelmed. The result is the same: uncontrolled lipid peroxidation, membrane rupture, and neuronal death. No single raindrop is responsible for a flood. Likewise, the trifold danger lies in relentless, overlapping convergence. A city commuter absorbs PM2.5 that can suppress GPX4, eats produce with trace organophosphates, lives in a sea of signals, carries low-grade inflammation or iron imbalance. The body has redundancies. The danger comes when different stressors target the same pathway. Pollution lowers firefighter numbers. Pesticides hide their tools. Frequencies spark more fires. Iron makes everything flammable. Years erode the shield, leading to a brain rusting from the inside out. This new understanding centered on GPX4 and ferroptosis is not despair. It is profound hope. A measurable problem is a manageable problem. We move from helplessness to strategy, focused research, decisive policy, informed personal choices. Measure GPX4 ferroptosis markers, link to exposures, and test ferroptosis blockers like ferrostatin 1. Remove chemicals that disable the shield, strengthen air standards, study EMF interactions, and protect vulnerable groups. The challenge is immense but no longer invisible. Work toward long life with a sharp, healthy mind. By protection, not luck. Thanks for watching. Please leave your questions and comments below. Share this video with friends and loved ones who may benefit. Thank you again for your interest and interaction. Think about it. You'll live longer.